Welcome to the second lecture on protein structure. Now, before I delve into this lecture, I would like to highlight that at this point of time, you should have gone through the other lectures on amino acids as well as the first lecture on protein structure, because if you haven't gone through it, this lecture will not add to your knowledge. So please first listen to the lectures on amino acids and the first lecture on protein structure before focusing on this lecture. Now, what are we going to do in this lecture is to define what is tertiary structure. We will see how tertiary structure is stabilized. After we have done that, we will move on to quaternary structure. Again, we will see the different interactions that stabilize quaternary structure of proteins. Then we are going to look at protein folding. Of course, protein folding is a discipline on its own, so we are going to focus only on the basic concepts of protein folding. We will like to highlight some of the key players in protein folding. We'll try to understand the function of these key players. Again, we are not going to focus on the detailed mechanism of how these different players of protein folding help in the process of folding. And towards the end of the lecture, we will be focusing on disorders of protein folding, which are very important because in assessments generally on protein structure, this is the area that is stressed upon. So tertiary structure, quaternary structure, basics of protein folding, some of the key players in the folding process, and at the end, disorders of protein folding. Of course, you need to know here that the tertiary structure of the protein can only be formed after we have the primary structure and the secondary structure. So this is the third level of protein structure that is formed only after we have the secondary structure. So first primary structure, then the secondary structure, and then we have tertiary structure. So it's a sequential process, and that's the reason I highlighted in the first slide that you should listen to the first lecture on protein structure before you focus on this lecture. Now, how do we define tertiary structure? Before I go into the definition of tertiary structure, we need to revisit what is primary structure and secondary structure. Now, you have already listened to the first lecture, so I am pretty sure now that you are aware of the fact that primary structure is the sequence of amino acids in a polypeptide or a protein. The secondary structure is formed when 3 to 30 amino acid residues in the primary structure gets geometrically aligned. And we have talked about in detail alpha helix and the beta sheet, the two major classes of secondary structure. We have also looked at beta turn. And once we have all the secondary structure that is formed in a polypeptide, then we will get tertiary structure. So tertiary structure is defined as the overall three-dimensional arrangement of all atoms in a protein. So here we have the secondary structure. If I focus on this particular figure, and this is an alpha helix, and now this is the primary structure, part of which it's formed the alpha helix. And once all the protein has formed into the different segments of all the protein molecule has formed the different secondary structure, it will start giving us the tertiary structure. So in other words, tertiary structure represents how secondary structural elements or features like helices, sheets, bends, turns, and loops assemble to form what we call the domain. So if you see here on this part of the slide, this is the tertiary structure, and you can see here how the different secondary structural features like alpha helix, this is another helix, this is a loop here, they all assemble together and they form what is called the domain. And the tertiary structure also deals with how these domains relate spatially to one another. Means you need to now visualize the structure as a three-dimensional element. So if I want to see it in 3D, it will have a spatial orientation, and that is what tertiary structure is. As I told you previously, for protein structure you will need to require to refer to your textbook. Now I just mentioned a particular term called domain. What is a domain? So domains are the fundamental, functional, and three-dimensional structural units of polypeptides. And polypeptide chains that are greater than 200 amino acids in length generally consist of two or more domains. Now this tells me something. In the previous lecture we talked about the difference between peptide and a protein. So a peptide is something which is less or equal to 50 amino acids. So a polypeptide containing less or equal to 50 amino acid is called a peptide. Whereas a protein generally consists of more than 50 amino acids. So I can say that domains will be formed only in proteins. Domains are generally not observed in peptides. So this you can note down, that domains will only be observed in the tertiary structure of proteins, because you require more than 200 amino acids in order to form a domain. So this is very important. I want you to read this particular section from the book, and if you have any doubts, please let me know. Now at each level of protein structure we have highlighted what is the major bond that stabilizes the structure. In the primary structure it is the peptide bond. In the secondary structure it is the hydrogen bond. So what is stabilizing tertiary structure? Now there are different interactions at play when the tertiary structure is formed. The first one, the hydrogen bond, again. The second one, hydrophobic interactions, which will be formed only between proteins which are only between amino acids which are non-polar. Then we have the ionic bond, which will be formed only between amino acids that are polar, and the disulfide bridge, which will be formed only between two cysteine residues. So there are four types of interaction that are required for the formation of tertiary structure. Number one, the hydrogen bond. Number two, the hydrophobic interactions. Number three, the ionic bond. And number four, if that polypeptide has two cysteines, then it is the disulfide linkage or the disulfide bridge. So if somebody asks you what are the major types of interactions that stabilize tertiary structure, you need to identify all these four different types of interactions. So in case when we talked about primary structure, it's only one, that is peptide bond. When we talked about secondary structure, it is only one particular interaction that stabilizes it, that is hydrogen bond. But when we talk about tertiary structure, you need to mention 4 different types of interaction. And they are the hydrogen bond, the hydrophobic interactions between non-polar amino acids, the ionic bond between polar amino acids, and if the polypeptide has 2 cysteine residues, then it is the disulfide bridge. So 4 interactions are to be highlighted when you talk about tertiary structure. Same like domains, I will again ask you to refer to this section in your book. Read this section, Interaction Stabilizing the Tertiary Structure. And I have talked about the 4 types of interactions, but in the book they are discussed in much more detail. And when you are reading this particular section, I will urge you to make some notes. Now we come to quaternary structure. So if I have one polypeptide chain, I will get first the primary structure, which is stabilized by the peptide bond. Then I will get the secondary structure, which is stabilized by the hydrogen bond. And then I will get the tertiary structure stabilized by 4 different types of interaction. Now, in a protein that has one polypeptide chain, we can have only up to tertiary structure. But in the physiological system, we have proteins which have more than one polypeptide chain. That means there are multiple chains in these particular proteins, which are called subunits. An example is hemoglobin. For example, we have hemoglobin. Hemoglobin consists of 4 polypeptide chains. And these are the 4 polypeptide chains of hemoglobin. If you see here in this particular part of the figure, 1, 2, 3, and 4. So these are the 4 polypeptide chain. Now, if we have a protein that has more than one polypeptide chain, then these chains will interact with each other and give you what is now called the quaternary structure. We will not have a quaternary structure if a protein consists of only one polypeptide chain. Then the highest structural level that will be observed is only the tertiary structure. But if I have a protein which consists of more than one polypeptide chain, I will get another higher order of structure, which is called the quaternary structure. So quaternary structure is restricted to proteins that have multiple polypeptide chain. And I gave you an example, hemoglobin. There are other examples which you are not going to look at. I talked about hemoglobin because later during the lecture we have to look at a particular disorder which relates to this particular quaternary structure formation in hemoglobin. So quaternary structure defines the polypeptide composition of a protein for an oligomeric protein. Oligomer means more than one polypeptide chain, and the spatial relationships between its protomers or subunits. Each polypeptide chain is called a subunit, and the spatial relationship between these subunits defines the quaternary structure. When I talk about spatial relationships, I refer to the 3-dimensional orientation of the protein. Now let us look at all the structures or all the structural levels of proteins with an analogy that I like to talk about with regards to alphabets and paragraphs. So we have individual alphabets. Now these alphabets will join each other. Similarly, we have one amino acid which joins with another amino acid, and the second amino acid joins with the third amino acid by a particular bond called the peptide bond, and we get what is called the primary structure. Now, when these alphabets are orienting themselves into meaningful entities called words, we can draw an analogy to secondary structure where 3 to 30 amino acids are getting a specific geometrical orientation. And this gives us the secondary structure. Two types, alpha helix, beta sheet. In formation of both the types, you require the participation of hydrogen bond. Now the words that are formed here group together to give you meaningful sentences. Similarly, secondary structures, they start interacting with each other and you get the tertiary structure. And then in proteins, or if I take a sentence and join it with another sentence and then a group of sentences forms a paragraph. Similarly, in proteins with multiple polypeptide chains, the polypeptide chains after forming the tertiary structure will interact, and this gives you the quaternary structure. So if I ask you what is the minimum number of polypeptide chains required to observe quaternary structure, the answer is 2, because a single polypeptide chain will give you structural level up to tertiary structure, but not quaternary structure. In order to form a quaternary structure, you require at least 2 polypeptide chains. And this is what you should note down while listening to this particular lecture. I would like to focus a bit more on domain. So we talked about domain. We talked that only proteins can form domains. So if I want to define a domain is a section of the protein structure sufficient to perform a particular chemical or physical task, such as binding of a substrate or other ligand. And most of the domains are modular in nature, and they are contiguous in both primary sequence and three-dimensional space. And this will become clear in the next slide. So let us look at a particular protein which is called triose phosphate isomerase. Why I took this protein? Because in your course you will encounter this protein. So this protein is a biological catalyst which is defined as an enzyme, and this particular protein has a particular domain that binds to 2-phosphoglycerate. Now if you look at it here, this is 2-phosphoglycerate, and this particular three-dimensional protein structure that you are seeing is the domain, but the actual protein is much larger. So again, this is a particular protein, triose phosphate isomerase. This particular protein binds to a particular chemical molecule, 2-phosphoglycerate, and only a particular properly folded part of the protein binds to the substrate, and that particular part of the protein is the domain. So this particular part which you see, which has alpha helix, some beta sheets, some loops, this is what is the tertiary structure, which is giving rise to a domain, and this domain has a unique function of binding to a chemical molecule called 2-phosphoglycerate. This chemical molecule is what people refer to as the ligand in general. We take another example, lysozyme. It's also an enzyme that is present in the cell in a particular organelle called lysosome. And lysosome contains this enzyme lysozyme. Of course, lysozyme is a big protein, but a particular part of the protein forms a defined tertiary structure which binds to a substrate called penta-N-acetyl pentaose, and this is the particular ligand or the substrate shown here in red. You don't need to remember any of the substrates. I just wanted to show you that only a particular part of the protein is involved in binding to a ligand, or more precisely, a domain has a definite function. For example, this particular domain in triose phosphate isomerase binds to this particular ligand. Similarly, this particular domain in the protein lysozyme binds to a particular ligand. Now, as I told you, most domains are modular. Now, if I look here very carefully, I have two domains. This is domain 1, which has a definite function of binding to one substrate. This is domain 2, which has a definite function of binding to another substrate, shown here in blue, and the substrate is shown here in red. So this is one module, module 1, and this is another module, module 2. So a protein may consist of more than one domain. So this is explained here with the particular protein, again very important in your course, lactate dehydrogenase. It's an enzyme, and you can see here it has two defined modular units, and these modular units are designated as domain 1 and domain 2. Domain 1 binds to a particular substrate, shown here in red. Domain 2 binds to another specific substrate, shown here in blue. As I told you, you do not need to focus on the name of the substrates, but what I want here is for you to remember that a protein may have more than one domain. So here the protein is having two domains. Similarly, a protein, if it is consisting of many amino acids, let us say 600 or 700 amino acids, it can have 4 or 5 domains inside that particular protein. And sometimes a particular domain is used as a signature to identify a specific group of proteins. For example, here the protein is dehydrogenase, and here we have a particular domain that is observed in the cell only in proteins which are belonging to the dehydrogenase family, and this fold is called, or this domain is designated as a particular fold. For example, this is defined as the Rossmann fold. So what is the idea here is for you to remember that a particular domain may be used as a signature for a specific family of proteins. Coming back, I talked about dehydrogenases. So dehydrogenases is a family of protein that participate in oxidation reactions, and in these proteins you will observe this particular domain, and therefore this particular domain is designated as a fold known as the Rossmann fold. You don't need to remember the name of the fold, but you need to know that a domain can be used as a signature to identify a group of proteins. Now, I mentioned that in the previous slides when I am showing the examples that the domains are binding to substrates or ligands, but not all domains will bind substrates. For example, there are certain domains called hydrophobic membrane domains which anchor proteins to membranes or enable them to span the membrane. Similarly, there are certain domains which regulate the function of other domains, and they are called regulatory domains. Regulatory domains trigger changes in protein function in response to the binding of allosteric effectors or covalent modifications. This is explained here. You see here, this is a particular protein that participates in the process of transcription. We talked about transcription, DNA to RNA. So this is a particular domain which is called the DNA binding domain, and this is a particular domain that participates in interacting with other proteins, so protein-protein interaction domain. So combining domains or domain modules provides a facile route for generating proteins of great structural complexity and functional sophistication. Let us take another example. This is the epidermal growth factor like protein. So these are schematic representations of the proteins shown here, where we have just focused on what we call the domain map of the protein. So this is a receptor called epidermal growth factor like receptor. This is a protein that has more than one domain. So you can see here, this is one domain, this is second domain, this is the third domain, and this can be regarded as the fourth domain. So in epidermal growth factor like receptor, we have a regulatory domain. The regulatory domain controls the activity of the catalytic domain, which is responsible for performing a specific catalytic reaction. Then we have a third domain that interacts with other proteins, so protein-protein interaction domain, and this is the transmembrane domain that interacts with the membrane or anchors the receptor to the membrane. Each domain has a specific function, and the question now comes is that if I remove one domain, will it retain its function? The answer is yes. If the folding is retained, each domain will retain its function. So if I cut this domain out from here and I retain the fold, then this particular domain will still interact with the target protein. If I cut this domain out from here, so domain 2 is taken out and the fold is retained, that means the tertiary structure is retained, then this particular domain will still participate in catalytic reactions. So I believe through these slides you now understand the definition of a domain and the fact that proteins may consist of multiple domains and each domain has a designated function, and this function can be retained even if the domain is separated from the other part of the protein and the domain has a defined tertiary structure. So we have here a tertiary structure of a protein which is called the three-finger proteins. So you have finger 1, finger 2 and finger 3. Each of these fingers represent one domain, and if you see here, this particular domain orientation or domain map is conserved across all three-finger proteins. So if you look at this one and this one, of course the spatial orientation may slightly vary, but you still have the three domains. Here also the spatial orientation may slightly vary, but you still have three domains. And this is a tertiary structure of a protein that has only one polypeptide chain. So you have anti-parallel beta sheets, and here you can see, although not very clear, these are the disulfide linkages that are formed between cysteine residues. So presence of a particular domain indicates a specific protein family. You can use it as a signature to identify a specific protein family. Here, the orientation of these three domains tells me that this is the three-finger protein family, and all proteins in this family will always have three domains spatially arranged like the three fingers of our hand. So which of the following best describes a protein domain? Of course, these kind of questions are rarely asked, but I just put it here so that you can test your knowledge at this point of time. And without going through the options, the correct option here is B, which actually tells you the definition of the domain: a discrete region of polypeptide chain that has folded into a self-contained three-dimensional structure. So this is what defines a domain. As I told you, these types of questions are rarely asked, but I just put it here so that you can test your knowledge when you are going through this lecture slide. So we come to quaternary structure again. Just to revisit it, I mentioned that quaternary structure will be observed in proteins which have more than one polypeptide chain. That means if I need to have a quaternary structure, I will require at least two polypeptide chains, because without that we cannot have a quaternary structure. If I have only one polypeptide chain, I will get only tertiary structure. I come to this concept map over and over again. In all proteins, we will always have till the level of tertiary structure. In proteins which have more than one polypeptide chain, we will always get quaternary structure. So with one polypeptide chain, the order ends here. So primary, secondary, tertiary. With more than one polypeptide chain, you will always get quaternary structure. I hope this concept is clear, because in the exam, questions just what I mentioned, for example, what is the minimum number of polypeptide chains that is required in order to observe quaternary structure? The answer is 2. We come to the next part. So we have finished protein structure, but what helps in the formation of this structure? What helps to fold the protein? Before we understand what helps to fold the protein, let us first understand how do we unfold the protein. Unfold the protein meaning, I take quaternary structure, I break the bonds between the different subunits, and here I will like to mention what are the bonds that holds the quaternary structure together. So if I have more than one polypeptide chain, what can be the bonds or interaction that hold these two chains together? The answer to this is very simple. It's the same interactions that you encounter in tertiary structure. So hydrogen bonding between the two polypeptide chains, hydrophobic interaction between nonpolar amino acids but now located on two different polypeptide chains, ionic interactions between polar amino acids located on two different polypeptide chains, and if the polypeptide chain has an unpaired cysteine, then it will form a disulfide bridge with another cysteine on the other polypeptide chain. So the interactions that you observe in tertiary structure will also be observed in quaternary structure. Now, if I want to break these interactions, then I will slowly start unfolding the protein. So the quaternary structure will give rise to tertiary structure. Tertiary structure will give rise to secondary structure. Secondary structure will give rise to primary structure. So if by some means there is a loss of three-dimensional structure sufficient to cause the loss of function, then that process is called the denaturation of a particular polypeptide chain or a protein. Now, how can we unfold or denature a protein? The first agent that is often used in the lab for unfolding or denaturing proteins is high temperature. As I apply heat, the interactions dissipate and I get to unfold the protein. Or the presence of chemicals such as high salt and reducing agent. What does salt do? Salt will interfere with the ionic interactions. Reducing agents will break the disulfide bond, converting the disulfide linkage to a thiol group. If you remember your lecture on amino acids, we spent considerable amount of time on this particular aspect. If I oxidize a thiol group, I get a disulfide bridge. If I reduce a disulfide bridge, I get a thiol group. Now, the question is, if I have unfolded a protein and then I put it back into the physiological system or in a system where there is sufficient or there is an ideal pH, ideal ionic condition, that is the physiological system. If I put the protein back into the physiological system, what will happen after I have denatured a protein? So this denatured protein, if it is returned to conditions in which the native conformation is stable, then it will undergo refolding. So primary structure, secondary structure, tertiary structure, and again I will get back the quaternary structure. There is one thing I will like to mention here. When I talk about protein being unfolded, I unfold it to the level of primary structure. I do not break the peptide bond, because if I break the peptide bond, then it's permanently destroyed. The protein is permanently destroyed. It cannot undergo renaturation. So let me reiterate. I take quaternary structure, convert it to tertiary structure. Tertiary structure is converted to secondary structure. Secondary structure is converted to primary structure. And I stop here. I do not break the primary structure into amino acids. I stop at the level of primary structure. So if I have a chain of amino acids and I put them back into conditions in which the native conformation of the protein is stable, then this primary structure will again refold and give you higher orders of protein structure. So this is an example shown here with a particular protein that has catalytic activity. These are the disulfide bonds shown here by the dashes between inside the polypeptide chain. So this is a protein that has one polypeptide chain. This is a protein that has one polypeptide chain. And what I do here is to add urea. And I will not answer why urea is denaturing the protein, because you have an exercise or a self-test that you need to answer, and there is a question, why does urea denature proteins? So I add urea and I add a reducing agent, mercaptoethanol. So what urea and mercaptoethanol do? First, urea unfolds the protein and mercaptoethanol reduces the disulfide linkages to thiol groups, as you can see here. Then what I do, I remove urea and mercaptoethanol, and the protein, which has unfolded in step 2, undergoes renaturation and refolds back into its native conformation. So this is a very, very schematic outline of denaturation and renaturation. Loss of three-dimensional structure is equal to denaturation. Regain of three-dimensional structure is renaturation. Now, what I described here can be done in a test tube. I can add urea, I can add a reducing agent, but inside our body, inside the cell, this cannot happen. So there is a different chemistry to it, and we need to look at some of the players in this folding process. So you get your protein chain. Protein synthesis happens from mRNA by translation. This protein is completely unfolded. If I keep this protein like this, it will quickly aggregate, and aggregates, that means it has completely a disordered structure, and these aggregated protein in general are toxic to the cell. So the body will remove, or the cell will remove, this aggregate. In rare occasions, this unfolded protein takes a very long time and gets folded into its native conformation. That means I get a tertiary structure if I have one polypeptide chain, or if I have more than one polypeptide chain, I get a quaternary structure. And then this particular folded protein is used by the cell for different activities. But this is not happening inside our body. What happens is, once we have the unfolded polypeptide chain, there is a group of proteins called chaperone, and these group of proteins assist the unfolded protein to fold and attain the native conformation, and this folded protein is then used by the body for different activities. So the first player in the process of protein folding inside a cell are a group of proteins called chaperone. What chaperones do is to assist in protein folding. Now the function of chaperone, if you want to refer to Wikipedia, or if you want to even refer to a book or material on protein folding, it's very, very elaborate. There are a lot of chaperones, and I will urge you not to refer to such material because it will confuse you more. Basically, chaperones are proteins that assist in protein folding. They take the unfolded form of the protein and convert it to a properly folded, native, or physiologically viable conformation. There are many proteins in the chaperone family. I will talk about two of them: HSP70, HSP, heat shock protein. H for heat, S for shock, P for protein. So HSP70 is a group of chaperones that binds short sequences of hydrophobic amino acids in newly synthesized polypeptides, shielding them from solvent, thus preventing aggregation and facilitating the formation of appropriate secondary structural elements and their subsequent coalescence into a molten globule or tertiary structure. So this is one of the very important chaperones. There are other chaperones like HSP90, and there is another family called the HSP60 family, which also assist in protein folding. But what the HSP60 does is that these family of chaperone proteins, sometimes they are called chaperonins, acts later in the folding process and assists in the folding of the protein. And they generally act together with HSP70. Why I mentioned HSP70 and HSP60? Because sometimes in the exams it is asked which of the following proteins assist in protein folding, and the answer, the options there will be an option either HSP70 or HSP60, or if they have the option chaperone, then you should choose the option chaperone. But generally they do not give you the option as chaperone, they give you HSP70 or HSP60. So we have covered one of the key players, chaperone. We have talked about two important chaperone proteins, HSP70 and HSP60. Now we go to the third player, that is protein disulfide isomerase. Now from the name itself you can understand that this is a particular protein that assists in the formation of disulfide bonds. But there is more to this particular protein. What protein disulfide isomerase does is it facilitates the formation of disulfide bonds between the correct cysteines that stabilize a protein's native conformation. Now, if I look at this part of the slide, we have again our favorite three-finger protein. The three fingers have three different loops, which I call as three fingers. So 1, 2, and 3. If I take this particular protein and unfold it, I will get first the domains, and then I will get the specific primary structure. But if you look at the primary structure here, you will see that the first cysteine is not forming a disulfide link with the second cysteine, but it forms a disulfide link with the third cysteine. Similarly, the second cysteine forms a disulfide link with the fourth cysteine. The fifth and the sixth cysteines are connected by disulfide bridge, so are the seventh and the eighth cysteines. So if I have to fold this protein in particular native or physiologically viable conformation, I need to pair the first cysteine with the third cysteine, not with the second one. So this correct pairing is done by this particular protein, which is called protein disulfide isomerase. So this protein not only helps in the formation of the disulfide bridge, but it also helps in the correct pairing of cysteines in a protein. So we have looked at chaperone, which help in the protein folding. We have talked about HSP70 and HSP60. Now we finished protein disulfide isomerase that helps in the correct pairing of cysteines and catalyzes the formation of the disulfide bridge between cysteines. Now there is one particular information that you need to know, that the enzyme protein disulfide isomerase, often abbreviated as PDI, requires riboflavin or vitamin B2 for its function. So if a person is suffering from a deficiency of riboflavin, there will be improper folding of disulfide containing proteins. That's very, very important. So if I have a cell system and in that cell I have proteins which have cysteine residues, and if this cell system is to generate properly folded proteins with correct matching of cysteines, it will require the presence of riboflavin in the physiological environment. Otherwise, there will be improper folding of the cysteine containing proteins if I take away riboflavin, because the function of protein disulfide isomerase requires the presence of riboflavin. Now we come to another important concept. Now when we have a protein, you think about try to picture in your mind a protein that has proline residues. We talked about proline in a bit of detail, both in amino acids as well as in protein structure, specifically in secondary structure. So if I am having a protein that has not undergone any folding, and this protein contains proline amino acid, and I have the X propeptide bond where X represents any amino acid. So I can put, let us say, isoleucine, proline, glycine, or X can be alanine, proline, leucine. So X represents any residue, then the proline will be in the trans conformation. But when we visualize X-pro bonds of mature proteins, approximately 6% of them are in cis conformation. And we talked about this, that proline is mostly present in a beta turn, and the cis conformation of prolines is mostly observed in beta turns. Isomerization from trans to cis is catalyzed by the enzyme proline cis trans isomerase. So coming back, when I have an unfolded protein, all the prolines will be in the trans conformation. But at certain positions in the protein, we need the proline to be in the cis conformation. And this particular trans to cis conversion is catalyzed by an enzyme which is called proline cis trans isomerase. Let us try to look at this. So this is a part of an unfolded protein that has proline residue. So this is the side chain, and this is the imino group, if you remember. So the imino group and the side chain are in the opposite conformation. So this is a trans conformation. Trans conformation is always favorable because it avoids the steric hindrance effect. Now, however, this particular part is in the beta turn, so I need to convert proline to its cis conformation. So this particular enzyme will convert proline to its cis conformation. Now if you see here, the imino group and the side chain are on the same side, not on the opposite side. So this is conversion from trans to cis. So this particular process requires the presence of a particular protein, which is an enzyme called proline cis trans isomerase. Now, why I wanted to talk about this in so much of detail? This particular enzyme or protein belongs to a family of enzymes known as cyclophilins. Now, cyclophilins not only help in the folding of physiological or native proteins, but also help in the folding of proteins expressed by viral invaders. So, cyclophilins are being actively sought as drug targets for virally transmitted diseases. So, if I can stop the function of proline cis trans isomerase, the viral protein will not undergo proper folding and therefore it will lose its virulence. A classical example of a cis-trans isomerase inhibitor is alisporivir, which is used for the treatment of HIV. So what this particular drug does, it goes and hinders the function or impairs the function of proline cis-trans isomerase. As a result, viral proteins cannot fold properly, leading to nullifying the effect of these particular virulent proteins. Before we move on, let us again revisit the different orders of protein structure, but this time we do so with the structure of hemoglobin. So hemoglobin consists of 4 polypeptide chains. So it has more than one polypeptide chain. So I will have quaternary structure in this particular protein. So it has 4 polypeptide chains. We have the 2 alpha chains and 2 beta chains. Mostly hemoglobin consists of alpha helix. That's the secondary structure. And then several of these helices, they undergo a specific folding to give you a spatial orientation. That's the tertiary structure. So this is one tertiary structure. This is another tertiary structure. And then all 4 polypeptide chains, after they have formed the tertiary structure, are linked together to form a spherical molecule. And this is the hemoglobin molecule. By the way, please remember that in case of hemoglobin, in the quaternary structure, we do not have any kind of disulfide linkage because there are no unpaired cysteine residues. So the interactions that stabilize the quaternary structure of hemoglobin are hydrogen bonding, ionic interaction, and hydrophobic interaction. So we will not have the formation of disulfide bridge in hemoglobin. Formation of disulfide bridge will only take place in proteins which have unpaired cysteines on their polypeptide chains. Now, that's the normal hemoglobin for you. Now we have a specific condition called sickle cell anemia. In sickle cell anemia, what happens is that there is an alteration in the genetic code. So this is the double-stranded DNA, guanine, adenine, guanine. So the complementary strand I think you all know this. G will pair with C, so cytosine, thymine, cytosine. Now, if I take the strand here and translate it, sorry, first I need to transcribe it to RNA. So what I will do here is to take this particular strand, so it's easy to deal with, and transcribe it. So C will transcribe to G, T will transcribe to A, and C will transcribe to G again. So that's the RNA strand. RNA, every triplet codon that we call, gives rise to one amino acid, and GAG gives rise to glutamic acid. Under certain conditions, the genetic code may undergo changes, and we call this change a mutation. So here A has been substituted with T. So this complementary strand will now have guanine will be complemented with cytosine, thymine with adenine, guanine with cytosine again. I take this particular strand for ease, transcribe it to RNA, I get cytosine to guanine, adenine to uracil, because we do not have thymine in case of RNA, and then we will have cytosine to guanine. So instead of GAG, we now have GUG, and this particular codon gives rise to valine. So glutamic acid has been changed to valine. A polar amino acid has been substituted with a non-polar amino acid. So polar to non-polar. This part, the translation and transcription process that I talked about quickly, you will become more familiar with it when you take your course on molecular biology. But what is happening at the protein level, for the time being, let us focus on the protein level. In normal hemoglobin, we have a glutamic acid. In sickle cell hemoglobin, we have a valine. So if I want to describe it more, in sickle cell anemia is a genetic disease caused by a single amino acid substitution from glutamic acid to valine in each chain of hemoglobin. So if you see here, you have a valine here, you have a valine here. So these are the 4 subunits of hemoglobin, the 4 polypeptide chains, and you have a valine here. What happens is, valine is a hydrophobic amino acid. So this particular valine that is present here will try to stay away from water. So it will start interacting with another valine on another hemoglobin molecule. So the hemoglobin molecule, which should stay apart, now they start forming a cluster, and the cluster formation takes place by hydrophobic interactions between valine residues or valine amino acids. And you can see here, this is shown in steps. So this is step 1. There is a complete cluster formation which have led to the formation of the strands, and then the strands have aligned to form the fiber. So what is happening here is that the valine has, the substitution of glutamic acid to valine has led to the clumping of hemoglobin molecules to form fibers. And this is extremely detrimental, because when the hemoglobin molecule is free inside the cell, the RBC is having a donut shaped structure. This is a typical human RBC, but when the hemoglobin molecule forms strand, their gradient density will increase, so they will start settling down on one part of the cell, as you can see here, and the cell becomes sickle shaped. Think of it that if you take a bag and put some weight inside it, the bag will bulge towards one side. That is the same thing that is happening here, because sickled hemoglobin, or hemoglobin with valine residues at position 6, will form fibers which have a very high gradient density, and then they settle on one side of the RBC. As a result, the RBC will attain a sickle shaped structure. And what happens is, generally the donut shaped structure of RBC allows it a lot of flexibility when it comes to its shape. But the sickle cell structure, because of the presence of fibers, has a sickle shaped structure, and this structure is not very flexible. So what will happen is, they will get stuck inside the blood vessels, as you can see here shown in this schematic. So the tissues will not get their required oxygen, because if they are stuck here, the RBCs cannot move to the different parts or the microcapillaries inside the tissues, and therefore the person will suffer from hypoxia. And since the blood vessels are stuck or blocked, there is excruciating pain in the person who suffers from sickle cell disease. In your study guide, there is a particular question that, especially in malaria prone belts, you will find people with sickle cell disorder, and I need you to answer that question for me, why it is so. So before we move on to the next disease, sickle cell disease is a typical example of a disease that happens when there is protein misfolding, because there is a change from polar to non-polar amino acid at position 6 in one of the polypeptide chains of hemoglobin. Now we come to the next set of protein misfolding disorders called prion diseases. So the word prion stands for protein infectious particle. And if you want to know the history of prion disease, it's kind of more of a modern disease that has been identified by a protein biochemist from Stanford University, Stanley Prusner, who was awarded the Nobel Prize for his work on this particular group of proteins. And what happens in a prion disease is that generally we have a bacteria or a virus or a fungi or some kind of living entity. Of course, virus is not a living entity per se, but we have some kind of nucleic acid involved in causing a particular disease. In prion diseases, the protein itself is the infectious particle. That's the reason it is called a prion disease, protein infectious particle. So in prion diseases, if I want to define them, these are fatal neurodegenerative diseases characterized by spongiform changes, astrocytic gliomas, and neuronal loss resulting from the deposition of insoluble protein aggregates in neural cell. As I told you, if a protein aggregates inside our body, it becomes toxic to the cell. And one such example of a prion disease, which is studied in detail, is the Creutzfeldt-Jakob disease. So what happens here? Let us try to understand. You need to now realize the fact that prion diseases, the cause behind them are not known and they are still elusive. So we do not know exactly what causes a protein to become infectious. So what I am telling you here is some from journals or reviews, recent reviews on prion diseases, but there is a lot of research going on prion disease, and still the disease remains quite elusive. So inside our body, we have the human prion-related protein called PRPC. Now, this is PRPC. If you see here, it is a protein that has a lot of alpha helix. So it's a helix-rich protein. Now, for some reason, this helix-rich protein suddenly gets converted to a protein that is rich in beta-pleated sheets. You can see here, these are the anti-parallel beta-pleated sheet. We talked about them. And this particular protein is then called PRPSC. Now the beta-pleated sheet, when you have a sheet-like structure, it increases the surface area of interaction for a specific protein. So these particular sheets start interacting with other sheets on other PRPSC. As a result, this protein will form aggregates, and these aggregates are insoluble and they lead to neural degeneration. Now, the most important thing is the PRPSC, which is rich in sheet with many hydrophobic amino acyl side chains exposed to solvent, and as each new PRPSC molecule is formed, it triggers the production of yet more pathologic variants in conformational chain reaction. So this will lead to another PRPC. So this PRPSC will go back and act on a normal prion protein, converting it to PRPSC, and so forth and so on. And then each PRPSC will interact with each other by hydrophobic interaction to form aggregate, and this aggregate is now toxic to the cell. Now the question is, there has been, unlike sickle cell anemia, there is no change in the amino acid sequence. It's just that for some reason, which we don't know, PRPC is converted to PRPSC. That means the folding is just getting changed. What causes the change in folding? Nobody knows for sure. There are many hypotheses proposed, but the exact cause for this change in folding is not known. So I believe it will give you an idea about what prion diseases are and how they are caused. So basically, we have a protein that has a lot of alpha helix. Then suddenly the alpha helical proteins gets converted to beta sheeted proteins. So predominantly alpha helical becomes predominantly beta sheeted. And once they are beta sheeted, the amino acyl side chains will start interacting and they will then aggregate, and the aggregated PRPSC or aggregated beta sheeted proteins are toxic to the cell. Moreover, this beta sheeted protein will interact with also alpha helical proteins, converting them to beta sheeted proteins. So that's the reason this particular form is called a prion or an infectious infectious prion. So prion, a protein infectious particle. We come to another neurodegenerative disease that is also caused by protein misfolding. And this particular disease, I think you have all heard about it. It's called the Alzheimer's disease. A lot of movies have been made on this particular disease, and it's a disorder that also remains elusive. Firstly, there are many people who do not believe in the idea that there is a disease called Alzheimer's. So, if you are interested, you can read a book called The Myth of the Alzheimer's. It is written by one of the top neurologists from National Institute of Health, and he believes that Alzheimer's is a disease that has been hyped up by the pharmaceutical companies in order to boost the sale of specific drugs. But whatever, what we need to know a bit about the disease. Again, the same concept of prion comes here. So we have something called beta amyloid protein. Now, inside our body, the beta amyloid protein is produced by proteolytic cleavage of a larger protein known as amyloid precursor protein. So beta amyloid is formed from amyloid precursor, and this is a beta amyloid protein, you can see here. Now this beta amyloid protein generally has generally in under normal physiological condition, it should have predominantly alpha helix. And what happens is that, again, due to certain changes in the conformation, the alpha helical protein is converted to a predominantly beta sheeted protein. You can see here, this is alpha helical, this is predominantly beta sheeted. The same concept of prion. And so if I want to summarize this, in Alzheimer's patients, levels of beta amyloid become elevated, and this protein undergoes a conformational transformation shown here, conformational transformation from a soluble alpha helix rich state to a state rich in beta sheet. Once we have this particular state, like prions, this will also lead to aggregation, and this aggregation leads to toxicity and neurodegeneration. Now, also this disease, as I told you, is very elusive, so people do not exactly know what is happening, why there is suddenly a conformational change. But certain theories tell that in the beta amyloid protein, which is generated from the amyloid precursor, the potassium at 28, the lysine at 28 position. The lysine K is abbreviation for the amino acid lysine at the position 28, somehow is responsible for the conversion, but nobody definitely knows if that is the correct mechanism. But people accept this, or let us say many people accept this as one of the reasons. Number two, the other thing that we should focus on is that there is a particular protein called ApoE or apolipoprotein E, which also has been implicated in causing the conversion of this alpha helical form to the predominantly beta sheeted form. But again, this is another school of thought that I am telling you, and I cannot, if you ask me, is this the cause, and I will have to say that I don't know. So either lysine or ApoE. So K is equal to lysine. It's a single letter code for amino acids. So either lysine or ApoE, or maybe both, are responsible for the conversion of alpha helical form to the beta sheeted form. But the idea here is once you convert the soluble alpha helix to an insoluble beta sheet, immediately you get aggregates, and the aggregated protein is toxic to the cell because it is prone to aggregation or self-aggregation, let us say, to be more precise. So we end this particular lecture with a take-home question. Which of the following orders of protein structure is most resistant to denaturation? It's not quaternary because you can denature it easily using heat, salt. Tertiary, the same. Secondary, also we can do the same because it's hydrogen bonding, so you can denature it very easily using heat. Tertiary, you can denature it using heat or any kind of salt that will take away the the electrostatic or ionic interaction, as well as certain salts nullify the hydrophobic interaction. The one that is most resistant is also primary structure, because primary structure, as I told you in the first lecture, involves the formation of peptide bond, which is a covalent bond, and covalent bond is a very, very strong bond. So the primary structure is most resistant to denaturation. Some of the concepts we will again revisit in our in-class sessions. So if you have any difficulty, I will like you to note them down, because certain concepts are pretty complicated sometimes when we suddenly start taking a jump from amino acids to protein structure. So my suggestion to you will be to take down notes during the lecture session, and if you have some clarification or if you have some questions, please talk to me during our in-class sessions such that others can also learn from your queries and questions. That brings us to the end of proteins and protein structure. And next we start with enzyme and enzyme kinetics, which is quite interesting. But before that, we will have a recap of these two lectures in our in-class sessions, followed by certain discussions, and also I will urge you to answer the self-practice test, which will help you to clarify more doubts. And if you have doubts when you go through these lectures, please send me an email or WhatsApp me your doubts. Thank you very much, and I wish you a nice day.